Hey guys, it's me Paola. Welcome back to another Bozeman video. Today we are going to be talking about the cell cycle. First though, let's just talk about life. For any living thing, there is inevitably going to be a point in their life when they die. Animals, plants, even cells go through this. So, the body has to have a way to replenish these cells. That's where this process comes into place. The cell cycle has many different steps that cover the process of how a cell makes new cells. Firstly, the purpose of the cell cycle is to create two genetically identical cells from one cell. The two cells are called daughter cells, as they came from the same parent cell. Most of the body and its organs go through this process of making new cells because some cells become damaged or may die, causing there to be a need for new cells to replace those harmed ones. The process of making new identical cells is known as a cell cycle. It is made up of three overall different stages: interphase, mitosis, and cytokinesis. As an overview, interphase replicates the cell's DNA or genetic material and prepares the cell for division. Mitosis divides the cell's genetic material in the form of chromosomes, and cytokinesis divides the cytoplasm, finally resulting in two daughter cells that have exact copies of information in DNA found in the parent cell. Interphase is the longest phase and has three parts: G1, S, and G2. In both G phases, the cell grows. In the synthesis or S phase, the cell's DNA gets replicated as the cell continues to grow. Because mitosis and cytokinesis are so short compared to interphase, they are usually just grouped together under the name mitotic phase, or M phase for short. There are many internal control checkpoints along this process though, acting as traffic lights to regulate and make sure that the cell is ready to move on to the next step. The first checkpoint is found in interphase in the G1 phase. Once a cell reaches a G1 checkpoint, there are two possible outcomes. The cell either gets a green light, allowing it to pass and complete the rest of G1 as G2 in the M phase and divide into two cells, or it gets a red light, where the cell exits the cell cycle and goes to a dormant G0 phase where it does not divide. The requirements for the cell to pass this stage are that it has to be large enough in size and have the necessary molecules needed for the next step. The next checkpoint is at G2 and is very similar to the first checkpoint. It makes sure that all the DNA has copied properly and is not damaged. If it is, then it goes to the M phase. If not, the DNA needs to be fixed, whether that means fixing damaged strands or making sure it gets duplicated correctly. The final checkpoint is the M checkpoint. It makes sure that the chromosomes or DNA that is now condensed into tightly wrapped coils are attached to the wire-like spindle fibers during metaphase. This is an important step because if the chromosomes are not all attached to the spindle fibers, then they won't be split up into two groups, and one of the daughter cells will have more chromosomes than the other, which can result in abnormalities. Preventing this is the exact purpose of the M checkpoint. If the chromosomes are not all attached, the checkpoint halts the cell from moving into anaphase and finishing the cell cycle until everything is in its place. The entire cell cycle control system is controlled by proteins such as cyclins and kinases. Cyclins initiate parts of the cell, specifically mitosis, to start. Kinases are dependent on cyclins, and they are enzymes that phosphorylate or add a phosphate group to proteins in order to activate or deactivate them. This cyclin-dependent kinase, or CDK for short, is present in all stages of the cell cycle, while cyclin has different forms or concentrations of itself present at the different stages as seen on the screen. Cyclin can attach itself to a CDK to send it to a specific protein with specific cell cycle locations and help it complete its job. Basically, both of these proteins combined, along with others, work to regulate the cell on a molecular level and ensure that the process runs smoothly. All of these checkpoints are controlled by both internal and external signals. For example, there is an external factor called maturation promoting factor, or MPF for short. MPF is present at the G2 checkpoint and it is made up of both cyclin and the cyclin-dependent kinases. MPF was first discovered in frog egg cells that were stuck in the G2 phase and were injected with a substance from cells in the M phase that allowed the first cells to continue the cycle. Cyclin combines with CDK to form these maturation promoting factor complexes that can be used to activate the M phase for a cell by adding a phosphate groups, as seen as purple, onto a target and this in the cell's nuclear membrane, allowing for the steps in the M phase to take place. Basically, once the CDK and the cyclin combine, it activates the cell and allows it to continue the cycle. Another signal used for these checkpoints is called platelet-derived growth factor, or PDGF. PDGF is made up of platelets, which are small groups, parts of cells in blood, and its purpose is to allow for blood vessels to divide and grow. It allows for stuck cells to pass the G1 checkpoint and thus be able to divide by sending signals. This is used by the body whenever an injury occurs, allowing for blood cells to divide quicker and heal the wound. The PDGF makes the blood cells divide, and these rush to a wound whenever an injury on the body occurs, helping it patch up and get fixed quicker, basically. However, sometimes this control over the cycles is lost. Some cells continue to grow and divide even when a complete layer is formed, just crowding on top of each other, or overall when there are not enough growth factors. These cells are cancer cells. These cells may continue to divide due to abnormalities like mutations in their control systems and the fact that the pattern in which they divide is random. Some cancer cells stop at strange points in the cell cycle rather than at the checkpoint, and others never stop dividing under the right conditions and with enough nutrients. Some normal cells, usually only dividing between 20 and 50 times in their lives before they die, transform into these immortal cancer cells that never stop dividing. Other cells have mutations that change the DNA and sneakingly pass right by the body's immune system, whose job is to usually destroy these insurgent cells, and continue to multiply and may form a tumor or a cluster of these mutated cells. On the screen is a picture of a tumor, and it has clusters of all the cancer cells invading the body. Some tumors, called benign tumors, can be removed because they stay in one area. However, others, called malignant tumors, spread across larger areas and affect different organisms or systems. This is known as cancer. Though there are some treatments to slow down the effects of cancer, many times it is impossible to completely cure it. All of this is due to the errors in the cell cycle control system that allow the bypass of these bad cells. Well, that's all for today's video, guys. See you in the next one!